first part of this lab, you'll be investigating the different cell structures found inside cells. And before we get into these different cell structures, let's first talk about the two main types of cells found in living organisms. Really, there's only two types of cells that are found in living organisms. One type of cell is called a prokaryotic cell, as shown here on the left. The other type of cell is called a eukaryotic cell, which is shown over here on the right. Prokaryotic cells are found in very primitive organisms like bacteria and another group of organisms called archaea that are very similar to bacteria. Eukaryotic cells are a lot more complex, and they are found in things like animals, fungi, plants, and a group called protists, which are also single-celled organisms like bacteria. However, they have more complexity in their structures. So let's talk about the major difference between prokaryotic cells and eukaryotic cells. Prokaryotic cells were the first cells on Earth. They have no nucleus at all. So if you look here at this picture, you can see, so typically the nucleus would be a nuclear membrane that surrounds the DNA. In this prokaryotic cell, you can see that the DNA is just floating inside the cell, and there is no membrane surrounding it. Whereas in the eukaryotic cell, you can see a membrane surrounding the DNA right here. Prokaryotic cells also do not have any membrane-bound organelles. Organelles, this term you will see a lot, just means structures within a cell. You can think of them as like tiny organs, and that's why they're called organelles. So organelles are these structures found in cells. However, prokaryotic cells do not have any organelles or structures that are surrounded by a membrane. Whereas if you look at this eukaryotic cell, you could see many membranes within the cell itself. Here's one. Here's another structure with a membrane surrounding it. Another here, 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 and so forth. So the term prokaryotic, pro means before, karyo means nucleus of a cell. So this word means before the nucleus. Eukaryotic cells, on the other hand, are a lot more complex. They have a nucleus, so this nuclear membrane surrounding the DNA, and they have membrane-bound organelles. So the structures, or in other words, organelles within the cell, have a membrane surrounding them. This gives it compartmentalization. Eukaryotic, eu means true, karyo means nucleus, so eukaryotic cells have a true nucleus. So let's talk about some more of the characteristics that differentiate prokaryotic cells from eukaryotic cells. Prokaryotic cells are smaller than eukaryotic cells, as you can see in this image here. So bacteria are always going to be smaller than any of the cells found in your body. Prokaryotic cells do not have any membrane-bound organelles. Like eukaryotes, they do, however, have structures called ribosomes, and these are these little dots that you see in here. And you can see those dots also in eukaryotic cells. These little dots are called ribosomes, and these are the sites of protein synthesis. So proteins are actually made on these ribosomes. Both prokaryotes and eukaryotes have ribosomes, which means that all cells on Earth have ribosomes. And one thing I just want to point out is that ribosomes do not have any membranes surrounding them. These are just little structures. They're actually made up of protein and RNA, and they float around in the cell and churn out proteins. Another characteristic that's interesting in prokaryotes is that they, unlike our DNA, which occurs in chromosomes, which are little strands of DNA, we have 46 strands of DNA in our cells. So we have 46 chromosomes. Whereas the chromosome, or in other words, DNA of a prokaryotic cell, is actually circular. So they have one large circular strand of DNA, and they also have these accessory pieces of DNA called plasmids. So these tiny little circles of DNA are also floating in here. If two bacteria cells come in contact with each other, sometimes they can actually transfer these plasmids between each other and therefore transfer genes back and forth. We'll talk a lot more about plasmids in a later lab. So let's focus mainly on the structures found in eukaryotic cells. And we're going to use two examples, animals and plants. And you want to be able to identify the structures in animals and plants, know what those structures do, and know whether these structures are found in both animals and plants, only animals, or only plants. Let's first start with a structure that is shared in both animals and plants. Over here on the left, I have a picture of an animal cell. Animal cells tend to be more circular or may have large variations in their shape, whereas plant cells tend to be very rigid and rectangular usually. And first, let's look here. Here we have the plasma membrane. So we're looking at this membrane that surrounds the cell. It's shown in blue over here. And there you can see this very skinny membrane surrounding the plant cell on the right. The plasma membrane is also sometimes called the cell membrane. In plants, however, surrounding the plasma membrane is a rigid wall called the cell wall. The cell wall is not present in animal cells. Since plants have cell walls, they have a lot of protection surrounding their cells. Whereas animal cells, since they do not have a cell wall, they don't have that protection, but it allows animal cells to be a lot more flexible. So what's the difference between the plasma membrane and the cell wall? Well, the plasma membrane, it's a very flexible layer, and you can see it right here. This is actually the cell membrane of one cell right up here, and this is the cell membrane of another cell on the bottom. The plasma membrane regulates what goes in and out of a cell, so we call this a selectively permeable barrier. It allows some things to go through, but not other things. Whereas the cell wall is very, very rigid. Here's a close-up of the cell wall of a plant. So it's a very rigid protective layer, and as I mentioned previously in the last lab, the cell wall is actually made of cellulose. So it's very, very fibrous. The plasma membrane, which again remember is found in both plants and animals, is composed mostly of two different molecules. The main structure of the plasma membrane are these phospholipids, which again are a type of lipid. They have a phosphate head, which means that there's a molecule containing a phosphate on one part of it. And you can see that as little balls here. And then attached to that phosphate head are fatty acid tails. And these fatty acid tails are nonpolar. Also embedded within the phospholipid bilayer are proteins. Proteins have a lot of roles in the cell membrane, but most importantly, proteins help transport substances across the cell membrane. There are some things that can't squeeze through the plasma membrane, so they require protein channels to bring them in and out of the cell. So now let's talk about the organelles within a cell. So remember, organelles are structures within the cell that have their own specific function. Many are surrounded by their own membrane. Here, surrounding the DNA is our nucleus. And if you look within the nucleus, you'll see this dark stain region. And that region is called the nucleolus. So don't get these two terms confused. The nucleus contains the DNA, as you can see here. However, here's a microscope image. So here's the nucleus, which I am tracing. And inside there, you can see this dark stained region. And this is present in every nucleus, and it is called a nucleolus. This dark spot is where ribosomes are made. And remember, ribosomes are the protein synthesizing factories within a cell. So ribosomes are actually made within the nucleolus, which is in the nucleus. However, these ribosomes will move out into the area surrounding the nucleus. Ribosomes, so what do they look like? Well, these things actually are made up of two different subunits, and these are two large proteins. And also, not really shown here, but there's also some RNA that makes up these two subunits. And here, shown in blue, is the actual protein that's being made, and it's actually a long chain of amino acids that's being built within the ribosome. Whenever you're looking at an image of a cell, ribosomes are usually illustrated just by drawing little dots. So you can see some of these dots are just floating around in the cell, and some of them are attached to this structure over here. Same thing in the plant cell. You see some ribosomes just floating around, and some of them are attached to this organelle over here. So these ribosomes are made, again, in the nucleolus, and they will move and make their way out of the nucleus. So let's talk about this structure here that looks very rough because it has all these ribosomes all over it. This structure is called the rough endoplasmic reticulum, or in other words, a lot of times we just call it the rough ER. Since the rough ER has a lot of ribosomes on it, it actually also plays a role in protein synthesis. It actually transports the proteins once they're made, and it transports them within the cell. Here you can see some images of the rough ER with the ribosomes all over it. This other structure here that looks a lot smoother is called the smooth endoplasmic reticulum, or the smooth ER. The smooth ER makes lipids. So this is where the lipids are synthesized in the cell. Remember that some lipids include things like fats, oils, phospholipids, steroid hormones. So all of these are made within the smooth ER. And the way that you can recognize this structure is that it's smooth, unlike the rough ER. I like to think of smooth. When I think of the word smooth, I think of like fats and oils, how they're kind of smooth and slippery. So that's how I always remember that the smooth ER makes lipids or fats and stuff like that. We also see this other structure. It looks kind of like the rough and smooth ER, but what you'll notice is that there's these little things budding off the sides. And these little structures budding off the sides are called vesicles coming out here. So when you see this membranous structure with these vesicles coming off the side, this is the Golgi body. The Golgi body is sometimes referred to as the post office of the cell. This is where all the proteins and lipids have to go before they can be marked or tagged and then sent outside of the cell. So here is another picture of the Golgi apparatus, or in other words called the Golgi body. So its function is that it modifies and packages proteins, which were made in the rough ER, and lipids, which are transported and made in the smooth ER, and it secretes them from the cell using these little sacs here called vesicles, or transport vesicles. And you can actually recognize the Golgi again because you will see all these vesicles budding off of the sides. So here's an actual microscope image of the Golgi bodies, and you can see these vesicles forming on the sides of this organelle. So let's put this all together. So what happens here, if we're talking about the lipids, let's just look. So here we actually see the rough ER down here. You can see all the ribosomes attached to it. Next to it is the smooth ER. The smooth ER is making lipids. It's transporting those lipids in a transport vesicle. Those lipids are heading to the Golgi body. And then the Golgi body is tagging those lipids, maybe modifying them, and then it's going to transport them in a vesicle outside of the cell. And the same thing would happen with proteins. Proteins would be made in the rough ER. Then they would be sent to the Golgi. The Golgi would secrete them from the cell. If you think about this all as a factory working together, it's a lot easier to remember this information. Okay, let's move on to our next structure. So here we can see this vesicle within the animal cell. There's also one in the plant cell. This is called a peroxisome. So now we're moving on to organelles that actually digest things within the cell. So we have peroxisomes here. We also have an additional digestive organelle within animal cells only, and that is called a lysosome. Peroxisomes are found in both plants and animals, and their main role is to break down excess fats within the cell. However, they also produce a toxic byproduct called hydrogen peroxide in this process of breaking down fats. So you actually may be familiar with hydrogen peroxide if you've ever poured it on a cut in order to sterilize a wound. So in order to deal with this problem where the peroxisomes produce hydrogen peroxide while breaking down fats, they also produce an enzyme called catalase. And you will be doing an entire lab on catalase in the future. What this catalase does, what this molecule or enzyme does, it actually breaks down hydrogen peroxide into oxygen and water. So it can take this toxic byproduct and break it down into non-toxic compounds. Another role that peroxisomes have is detoxifying other toxic byproducts of metabolism. Lysosomes, on the other hand, these are only found in animal cells. They contain enzymes that digest macromolecules, so really large molecules. Not only do they break down large molecules, they also break down cell components that are no longer needed. So for example, if you look at this image here, you can see the lysosome, which is really large here. And this lysosome is actually breaking down and recycling a mitochondria within the cell. And it's breaking down a peroxisome. So it's actually recycling cell components that might have aged or maybe are not working properly. And as I said before, lysosomes are not found in plants, but plants do make similar enzymes that break down large macromolecules and stuff. And they make these enzymes in a structure called the large central vacuole, which we will talk about next. So if we look in both animal and plant cells, we have these storage organelles called vacuoles. However, if you notice in the plant has one large central vacuole, whereas in animal cells, there's just many tiny little vacuoles throughout the cell. What does a vacuole do? Well, it's a small sac that stores fluid, but not just water, it also stores nutrients and may store waste as well. Although the vacuoles are found in both plants and animals, plants have a very large central vacuole. This large vacuole stores water, nutrients, waste, and it creates water pressure, or in other words, turgor pressure within the plant cell. It also serves the same role as the lysosomes in that it breaks down cell components and macromolecules. So here you can see a plant cell. Here's the cell wall. The inner black circle is the cell membrane. And then we have another circle, which is the vacuole. If the vacuole doesn't have enough water in it, the cell wall will become very limp. And this is what causes a plant to wilt, because the vacuole is not creating enough water pressure inside of it. However, if the vacuole is filled with water, it can push out on the cell wall, and then your plant will be nice and rigid and tall. If you forget to water your plants, the reason they start to wilt is because the vacuoles are becoming empty. Next, let's talk about some organelles that create energy within cells. First, what we see in both animals and plants here, they look like little kidney beans. These are called the mitochondria. Mitochondria are known as the powerhouse of the cell because this is where most of the energy production occurs. And cells need a form of energy called ATP to do anything. And all the ATP that a cell needs is made in these mitochondria. So we say that they are a major site of metabolism. They allow our body to do things. One thing I want to emphasize is that mitochondria are found in both plant and animal cells. Both plants and animals need to make ATP, so they need the mitochondria. Also notice that within the mitochondria, we see all these folds. And that's an easy way to identify the mitochondria because they always have all these folds inside, as you can see here. These folds increase the surface area for ATP production. Now, if we look at plant cells, they actually have an additional structure here called the chloroplast. Chloroplasts are only found in plant cells, not in animal cells. Chloroplasts are the site of photosynthesis in plants. They're green because they contain a pigment called chlorophyll, and chlorophyll is actually the molecule that absorbs solar energy. Because chloroplasts are involved in photosynthesis, this is actually where glucose is made within plants. And here, if you were to look at a plant cell, I'm going to click here, and you can see that the plant cell is outlined in red. And in the middle here, any of that blank space is the central vacuole. And if I push play in this video, you'll actually be able to see the chloroplasts moving around within the cell. So these chloroplasts are organelles that we can actually see with the light microscope. And again, this is where photosynthesis is happening in a plant, and this is what gives plants their green color. If we look inside the chloroplast, it looks different than the mitochondria in that there's actually little sacs stacked on top of each other within the chloroplast. The purpose of these sacs is to increase the surface area for photosynthesis to occur. Finally, let's talk about the fluid within the cell. The fluid within the cell here, which is shown in blue over here and green over here, is called the cytoplasm. The cytoplasm is a fluid between the cell membrane and the nucleus, and it's where all of the organelles are suspended within the cell. There are a few other structures that we need to talk about that have an important function within cells, and one of these structures are these. Do you see these little lines in here? Well, these are like skeletal structures within a cell in a way. These are actually protein fibers, and we call these structures part of the cytoskeleton. It's like the skeleton within the cell. Cyto means cell, so it's a skeleton in the cell. The cytoskeleton, again, are protein fibers that function in both intracellular support. So here you can see all the cytoskeleton rods, and they're going to support the cell membrane. They also function in transport within eukaryotic cells. You can see these little blue structures here are actually moving along the cytoskeleton. So sometimes they almost serve as like highways for things to be carried from one place to another inside of a cell. The cytoskeleton is present in both plant and animal cells. A structure that you will see within the animal cell looks kind of like these two little churros. If you know what churros are, they're like little desserts that have these lines on them in a way. So these two little churro looking like structures are called the centrioles. They are only present in animal cells, and they're not present in plant cells. Centrioles are found within a region within the cell called the centrosome. The centrosome is the area or the region of the eukaryotic cells. So it's both plant and animal cells that have a centrosome where microtubule spindles, which are these fibers here, are formed during cell division. So centrosomes play a key role when cells divide. The centrosome, again, is the region here, this entire region. However, in animal cells, the centrosome contains paired centrioles. And we don't really completely know the role of the centrioles. We just know that they are present in animal cells, and they also play some role in cell division. So, recap. Centrosome is found in both plants and animals. It's the area that creates these microtubules or these protein fibers during cell division. However, the centrosome in plant cells does not have centrioles, but the centrosome in animal cells does have centrioles, which are those paired structures that look like churros. All right, so that's the end of part 1 of this lab, looking at cell structures.